Hi everyone. Today we will be discussing about chemistry and enzymology of DNA replication. So, what is a DNA? DNA is a long polymer of deoxyribonucleotides which carries the genetic information from generation to generation. And it is also a process in which DNA is multiplied, or let's say, in other words, it is copied itself to produce identical daughter molecules of DNA. So the collection of enzymes, actually many different kinds of enzymes are involved to produce the DNA. Um, and there are also many physical and chemical reactions that take place during the replication process. So DNA replication is primarily concerned with the making and breaking of phosphodiester bond, which is the backbone of DNA. So the enzymes involved are RNA primase. So what is RNA primase? It is a short RNA oligonucleotide segment. It is synthesized on the DNA template by a specific RNA polymerase called primase. So the constant synthesis and supply of RNA will occur on the lagging strand of DNA. So in bacteria, there are two different enzymes which are known to synthesize the primer. They are RNA polymerase and DNA primase. So this RNA polymerase, which has activity on the leading strand, and DNA primase on the lagging strand. Next enzyme we'll be talking about is DNA helicases. These enzymes bind to both the DNA strands at replication fork. So helicases move along the DNA helix and they separate the strands, which we all know. So their function is compared with a zip opener. So helicases are ATP dependent for energy. They utilize ATP for energy, which promote the separation and which creates the replication fork. So the next enzyme is SSB protein, single-stranded DNA binding protein. They're also called as helix destabilizing strand. So they destabilize the helix and they possess no enzyme activity. They don't act exactly like an enzyme, since that's why they're called protein, SSB protein. So during replication, the single DNA strand are prevented from rewinding. So once the strand is separated, they tend to rewind. So this rewinding is prevented by the SSB protein. It is believed that the SSB protein, they also protect in the degradation of the single stranded DNA by nucleases. Topoisomerases. So what are topoisomerases? So whenever there's a supercoil which happens in the during the DNA replication, this topoisomerase enzyme helps the DNA strand to unwind or not get coiled. So that is when the replication will proceed. So there are two types of topoisomerases. They are DNA topoisomerase type 1, DNA topoisomerase type 2. So type 1 cuts the single DNA strand, and type 2 cuts both the strands and reseals them to overcome the problem of supercoil. DNA polymerases is the next enzyme. So there is a lot about prokaryotic DNA polymerase and eukaryotic DNA polymerase, but here it's given in very short information. So prokaryotic DNA polymerase is DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. So DNA polymerase 1, it is a DNA repair enzyme, and it is involved in removing the RNA primers from Okazaki fragments. And DNA polymerase 2 resembles the DNA polymerase 1. It's just as the same like DNA polymerase 1 in its activity, but it's a DNA repair enzyme. Both are DNA repair enzymes only. DNA polymerase 3 enzyme plays an important role in DNA replication. It is also known as a hollow enzyme or multi-enzyme complex having 10 subunits. In eukaryotic DNA polymerases, there are five types: alpha, beta, gamma, delta, epsilon. DNA ligases DNA ligase enzyme. So these enzymes, they are basically uh, capable of catalyzing the phosphodiester bond, which is formed in between the 3' OH and the free um, 5' phosphate groups of a nick of the DNA. So it is used to link or ligate or glue the fragments of the DNA together. So it is used both in DNA repair and DNA replication. Telomerases. So it is an usual enzyme as it is composed of both protein and RNA. So the telomerase RNA it base pairs to the end of the DNA molecule. By the end of the DNA molecule, it will base pair, and the telomers they help to extend to a small distance. Then the telomerase translocation occurs and extension happens. So the process of DNA synthesis and translocation is repeated several times until the chromosome gets sufficiently extended. We all know that there is a protein called telomere on the ends of the chromosome. The extension process gets completed through the participation of DNA polymerase and primase, sealing the new DNA which is formed. Thank you.